Yeah, welcome here everyone in the third week of Bioinformatics for School Children, fourth week of Bioinformatics for School Children program. Here today we have with us Dr. Aparna Banerjee. She had her Ph.D. in botany from the University of Bern, India. She has her bachelor's and master's degree in biotechnology, and she has done her postdoctoral research at University of Maurya, Chile, where she is currently working as assistant professor for last more than 4 years. And in her 8 years of research experience, she has worked on applied microbiology of extremophiles and microbial bioactive compounds. Her research interest also includes biodiversity, conservation, and bioinformatics. She is the principal investigator for four different Chilean national funds and Chilean Antarctic Institute Fund that deal with bacterial polysaccharide produced by thermophilic bacteria and their applications as food additives. She is also co-investigator in four other Chilean national and international funds. She has more than 45 publications in nationally and internationally reputed journals and has been an awardee of National Science Congress Association Young Scientist Award and Swan Jayanti Puraskar. So we are very much honored to have you with us here, ma'am. Please continue the session.
Thank you, Rajul. Thank you for the introduction. So, as I told to the kids, if you find problem at any time, please feel free to stop me at any point so that we can keep it interactive. Can you see the slide?
Yes, ma'am.
Okay. So, my name is Aparna, and I'm working as assistant professor in a university in Chile, in another part of the world. I'm basically from India, but last almost 5 years I'm in Chile. So this country probably we almost all know for football, but other than that, Chile have a real nice similarity with India for the geography. If we think in India, the northern part of the India Himalaya is extreme cold, and the southern part is very hot and humid. In Chile, it's kind of similar. The northern part is very hot, and the southern is very cold. Welcome to today's talk. I'm going to talk more into the part of microbiology in bioinformatics. Bioinformatics is a very interdisciplinary field. So, what is the position of microbiology and how we can understand microbiology through the help of bioinformatics? That's something that we will talk today a little about microbiology and a little about some small use of bioinformatics and microbiology. The next class we will have in more details, but today it will be based on basically some introduction and a few hands-on session that you will work with me also. Together we will do it. So how this term bioinformatics came? The term bioinformatics came in 2001. There was a beautiful publication that talks the application of computational tools to organize, analyze, understand, visualize, and store information associated with biological macromolecules. So often we have, for this reason, often we call it as computational biology also, to understand biology with the help of computational tools. The term bioinformatics was coined in 1970s by Pauline Hodgweg and Ben Hesper, and they describe bioinformatics in another way. You can see in the left Pauline and in the right Ben Hesper. So they described it the study of informatic process in biotic system, thus bioinformatics. That's the way they described it. It is also referred as computational biology. However, computational biology deals more with the modeling of biological system. So but the main component of bioinformatics are developing software, developing tools, new algorithms, and analyzing and interpreting the biological data using those variety of tools, softwares, and algorithms. So in bioinformatics, we are having, it's a total interdisciplinary field where from mathematicians to statistician to biologist to chemist to physicist work together to understand a particular system from different point of views. Now, after bioinformatics, what are microbes? The term microbes we often confuse with virus, with bacteria, but other than that, there are a lot. Archaea, probably we have not heard it before, but this organism, Archaea, is also a microorganism and that normally lives in extreme environment. fungi are also microorganisms. Sometimes the macrofungi like mushroom, they are not microbes, but the microfungi, unicellular organism, are microorganisms too. There are two types of algae, macroalgae and microalgae. The macroalgae we can see, but the microalgae we cannot see through our naked eye, nostoc, anabina. So they are also microorganism. The protozoa like amoeba, they are microorganism, and viruses definitely. So there are different kind of microorganisms, but the question is, where are microorganisms? Where they live, isn't it? So they live in almost all possible environment. In plant, in water, in air, in plant where they live, inside the plant they live, in the root rhizosphere, they make a symbiosis with the root and then they live. In this kind of symbiosis, they help the plant to get some nutrients, and in order they survive inside the root of the plants. So basically, they help the plants to grow. In case of our body, we have symbiotic Escherichia coli, we have lactobacillus. The microorganisms are living in our body, helping us to grow, helping us to have a healthy life, and in order they are surviving inside our body. And if this is a room, and in the room we cannot see where is air, the microorganisms are present over there too. And of course, in water, there are a lot of microorganisms. So microorganisms can be two types. One is free living and one is associated. The free living is in the air, in the water, separated, and the associated are making microbiome. They are either associated with a plant, with animal, with other organisms, and making a microbiome. So now the concept comes. When we think about microorganisms, they are bad. And this concept more came after COVID, isn't it? When we hear the term microbe, we fear. But that's not the case. If we think 100% of microorganism, probably 1% of them are pathogenic and 99% are non-pathogenic. Often they are non-pathogenic, but when we call them good bacteria, let us imagine our body. Some bacteria cause pathogenicity, they are bad bacteria. But there is, in our body, we have more than 10 times of our own cell that are bacterial cell. So in more than 10 times of our body's own cell are microorganisms that are living inside our body, and that is helping our body to get nutrients, to have a it's maintaining our body metabolic system. And whereas in comparison, the bad bacteria we can only understand when we are getting some disease. So this concept of good bacteria and bad bacteria is somehow that is getting portrayed in a way because disease is something like dangerous. But if we even forget about good bacteria inside our body, let us think about the world. A lot of microorganisms in air, in water, in our planet Earth are maintaining the biogeochemical cycle, carbon cycle, nitrogen cycle, phosphorus cycle. All these metallic cycles, gaseous cycles are maintained by the microorganism. So imagine now in this time of climate change, in this time of global warming, if we lose an ecosystem, we will lose that microorganism. If we lose one microorganism, there will be a irreversible change because that biogeochemical cycle will be hampered. So we need to know about the good bacteria too in a time when we are focused much about the bad bacteria. So microorganisms are basically omnipresent. In all the environment where we cannot think about life, microorganisms are present. So often, to study the analogs of space, to study extraterrestrial life, and to make a mimicry of that, we study microorganism in extreme environment. Can be polar region, the permafrost, mud volcanoes. Mud volcano we have in India also, in Baratang mud volcano near the Barren Island in Andaman Nicobar part. Hydrothermal vents, hot spring, fumaroles, volcanoes, desert and arid environment. For example, Thor Desert, some acid mine drainage, anoxic lakes, deep sea hydrothermal vent, soda lake. Soda lake we also have in our country. The Lonar Lake in Maharashtra is a soda lake. It's a hypersaline lake. Some nuclear contaminated sites. So in all this extreme environment, in very high temperature, very low temperature, very high pH, almost zero pH, to very basic pH, to very high salinity. A lot of heavy metals and trace metals are present. A lot of ultraviolet radiation is present. More than 1 atmospheric pressure. In all these possible extreme environment where life is not present, microorganisms are present. If they are present, how we come to know who is the microorganism? Like, for example, my name is Aparna. How we came to know who is bacteria and what is the name of that bacteria? That is the part where bioinformatics plays the key role. For the identification and for the nomenclature of microorganism, bioinformatics is the rudimentary step. For plants, for animals, seeing their morphology, we can determine them. But in case of microorganism, no. A lot of bacteria are bacilli. Actually, bacilli is the most complicated and most densely populated microbial group, bacterial group, genus. So how we can understand which particular bacilli it is? There we need the help of bioinformatics to understand microbiology, the very basic of microbiology, which bacteria it is. So how can we see and identify a bacteria when they are so small, isn't it? The macro life is very easy to understand. There is plant, this plant is hibiscus, that plant is mango, that is a jackfruit, isn't it? That is a lychee. Or in case of, even in case of some animal, we know, okay, this is a penguin, that is a cow, that is a crow. But in case of microorganism, we cannot see it through our naked eye. And if we cannot see it through our naked eye, how can we identify them? Isn't it? Till now, is it okay for all the kids? Like, because I cannot see you all, I hope till now it's okay.
Yes, ma'am.
Okay. So the very basic technology to understand microorganism was developed by Hans Christian Gram. This man developed a method of staining to distinguish and classify bacterial species into two very large groups. One is gram-positive bacteria, one is gram-negative bacteria. And the name comes from the Danish bacteriologist Hans Christian Gram. He developed this technique. So what he did, he takes some stains, crystal violet, a violet colored stain, and saffronin. This stain is a pink colored stain. So in a simple microscope, this microscope that you are seeing, it's a compound microscope with a light connection. In probably in our school or something, we will have a without light connection, simple compound microscope. If we take the microscope near the window and the light source is there, sunlight is coming, we can see the bacteria clearly on that. So when we stain, for example, we take a little water from the tap, or we take a little soil, we dilute it, and we put a drop in a slide, we can see something like this. Some violet gram-positive bacilli or some rod-shaped bacteria, and some pink gram-positive bacilli or some rod-shaped bacteria, isn't it? So why few take violet? Why few take pink? The first we give is, in the sample, we give a drop of crystal violet. So when you give the drop of crystal violet is very hard. Don't penetrate the bacterial cell. We give some grams iodine. It acts as a mordant. And then we try to wash them, and then we put some saffron in. The microorganism that have a very thick cell wall, there the crystal violet enter and don't go out while washing. And in case of bacteria that have very thin layer of cell wall, when you wash, the stain go out and the saffron remains. So in basically identifying the composition of the cell wall of the bacteria, it's gram-positive bacteria that have thick cell wall and gram-negative bacteria that have thin cell wall. But then comes the problem. For example, Mycobacterium tuberculosis. Don't stain by gram stain. It gives false gram staining. Why? Because Mycobacterium tuberculosis is a mycolic acid. It's like a wax. So if you heat it, then crystal violet enter. But when you don't heat the bacteria, crystal violet don't enter. Like wax, when we heat it, the wax melt. So then slowly, a lot of problems started coming, even with Clostridium, a lot of bacteria that basically don't get stains with gram staining. There comes microscopy. So initially we started seeing with a simple microscopy, compound one. Then comes scanning electron microscopy. The scanning electron microscopy help us to see the morphology of the bacteria, how they look from the outside. While in case of transmission electron microscopy, we can see the inside. It's all about electron, how the electron is penetrating and not penetrating. Electron microscopy works into that. So while in compound microscopy, light help us. In case of electron microscopy, electron help us to see. Confocal microscopy, in confocal, we see different levels, how bacteria are organized, and basically with some stains, some confocal microscopy stain. Atomic force microscopy, this microscopy helps to see the three-dimensional organization of the microorganism. So there are a lot of microscopical technique to see bacteria. So okay, you can see the bacteria, they are very small, you can see them. But how can you know which bacteria they are, isn't it? How to know them and how to identify a bacteria? There comes bioinformatics. Bioinformatics is the rudimentary step, and it's the torch bearer basically to identify a bacteria. You can only say a bacillus or a coccus or a comma-shaped bacteria. More than that, you cannot say which bacillus it is. To identify, we need to understand bioinformatics. So there is a technique called, let us not go into much deep, let us not think into too much details of it, but just to tell very simply, we all have ribosome, isn't it? We have ribosome in our body cell.
Yes, ma'am.
Which ribosome we have in case of eukaryotes? ATS, isn't it? I don't know if some of you have studied this part or not, but if you have studied, let us brush up. It's 8Ts, okay, in case of eukaryotes. So there is a large subunit and there is a small subunit, 6Ts and 40S. In case of prokaryotes, it's a 70S ribosome. S is for sedimentation, Svedberg unit, isn't it? So 50S is the large subunit, 30S is the small subunit. In this 50S, there is 23S, 5S and ribosomal protein. And in the small subunit, 30S, 60S and some ribosomal protein. This 16S ribosomal RNA gene codes for the RNA component of the 30S subunit, as I told. And this 16S ribosomal RNA is widely presented in all the bacterial species. All means all. So 16S rRNA gene sequencing is one of the most common method where we target the housekeeping gene. What is housekeeping gene? The gene that let us identify which bacteria it is. So for example, Escherichia coli have a particular 16S rRNA gene sequence, while Bacillus subtilis have different. All the Bacillus subtilis have same, all the Escherichia coli have same. A little point mutation can happen sometime, but the basic structure is same. That is the idea of housekeeping gene. So this 16S rRNA gene sequencing among a lot of housekeeping genes, it is more universal, that help to us to understand bacterial phylogeny, genus and species classification. So it assists with differentiating between closely related bacterial species. For example, Bacillus cereus and Bacillus anthracis, they are very close. Many clinical laboratory, they use this method to identify unknown pathogenic strain. For example, if there is an infection of tuberculosis, if there is a infection of anthrax, they try to understand which is the causative organism. They do sequence, and with the help of bioinformatics, they identify which pathogenic bacteria it is. According to the guidelines of bacterial classification, you have some control. And if with the control, your 16S rRNA is showing more than 97% similarity, then you can have an idea that they are similarly related. They are related a little similarly. But if it is showing less than maybe 97% similarity, then you have to go through alternative approach. There are different other alternative approach, hybridizing the DNA and all this, and that can be a novel species. But in case of more than 97%, we can know, okay, so this is the genus, but for the species, if it's 100% same, it's the same species. So this is the way With this sequencing method, we can identify a bacterium. And why we need to understand which bacteria it is? Because microbes we are having in our daily life. Even if we cannot see it, there is. If we think about food, probably we all love dosa, isn't it? Dosa, idli, curds, paneer, cheese. We all love these things or not. All these foods have bacteria, and they are good bacteria. They help in fermentation, and the fermented foods are rich in vitamins, rich in nutrients, rich in antioxidants, and often the doctors tell us to take more fermented foods when our stomach system is not okay and we want to increase our immunity. Often the doctors advise us to take fermented foods. So how they ferment, how the fermentation process goes on, microorganism does the fermentation. Let us think about waste. There are two kind of waste: solid waste and liquid waste. There is gaseous waste also, but gaseous waste we cannot see. In case of solid and liquid waste, regular treatment process include bacteria. Often the bacteria produce some kind of polymers that help flocculating into the particles. So if you have a big wastewater remediation plant and you put your bacteria that produce these polymers, they flocculate all the material, and all the materials, waste material, get precipitated and you get the water out. So we often use them in our daily wastewater treatment plant. In medicine, a very big concept that's going on nowadays is nutraceuticals. I don't know if you have heard about this or not. It's not pharmaceutical, it's nutrient and pharmaceutical together, called nutraceuticals. Let us think about Yakult. I don't know if you people have seen in India nowadays this kind of small shot of fermented milk, often the pharmacies sell, called Yakult. It basically have lactobacillus casei, and lactobacillus casei in a particular dose. So when we take the lactobacillus casei, it goes into our digestive tract. They divide and helps our body to get the necessary nutrients and hormones produced by them. And fourthly, in our rhizospheric microbiome, a very good example can be pea plant. Probably all of you have seen peas, green peas. If you take a green pea plant when grown, full grown plant out, you see their roots. Sorry, I have a little cough, so often I'm like making this, but in this time of seasonal change, I'm having this. Sometimes I'm missing the link. So in this kind of rhizospheric microbiome, if you take the plants out, you see the roots, and if you see small tumorous growth. What are those tumorous growth? These are not tumors. If you take those nodules out, you cut them and put it in a compound microscope, you see bacteria. So they are basically symbiotic bacteria. They make nodules in the plant. They help the plant to get protection against pathogen. They send some protective molecules. They help the plant to tolerate against abiotic stress. They produce a lot of compound that help the plant to grow in a very dry environment. They make a pseudo watery environment. Basically, they trap the water inside the root. They produce some compounds. They help the plant to grow and develop, help them to acquire nutrient, and help totally in the physiology and metabolism of the plant. So everyday life, if we are using them, how to know which bacteria to use? There comes this technique. The technique is 16S RNA genome sequencing that we told. So what we do, we isolate a bacteria. How can we isolate a bacteria? A very simple experiment can be, you take tap water, you put it in some media, formulated media where there is some carbon source, nitrogen source, and salt. When the colonies come, you isolate the bacterial DNA from them. You amplify the 16S RNA gene. Now, what is the word amplify? Probably you all know about PCR, isn't it? For COVID-19, probably you all have heard this term PCR. This PCR is called polymerase chain reaction. And a machine called thermal cycler help, if you have a DNA, it amplify the DNA into lot more quantity. So if you have one DNA, they can make it 100 of DNA. So anything is better when you have more quantity, isn't it? You are trying to identify. Definitely, to have less error or zero error, you need to amplify a very small portion in a larger portion. Then you sequence this large portion of the 16S rRNA gene and compare the sequence gene with GenBank to obtain a match. So from here, we will go to till this part. It's okay, kids? It's okay till this part? I'm pasting here. Just play with this link. You can open directly from the chat box this link. So this NCBI, National Center for Biotechnological Information, it's a database. This database have, you can see what this database have genome, have nucleotide, have protein, have all. So for example, you want to search a 16S rRNA gene sequence. Let us think, lactobacillus. Let me search from here only. Once a minute, the laptop got hanged. Okay. So let us search from this only. The example of the presentation, because the presentation I'm going to share with you also later. You can search with any bacteria, Escherichia coli, Bacillus subtilis, Bacillus cereus, anything. So this Bacillus licheniformis came. What you can do is you can submit your sequence in NCBI database. And NCBI database have a very nice tool. For example, 16S rRNA, Bacillus licheniformis 16S rRNA gene. You click here and there comes an option called Run Blast. What this blast do? The blast basically do compare your sequence with the database. So here I am just showing you with an example of the sequence that are already submitted in NCBI. But if you are sequencing a new bacteria and you know the genome sequence, you submit it in NCBI, you can run blast and can identify which bacteria it is. So what you have to do here, rRNA ITS database, bacteria and archaea, and blast. It will take some time, but will come. There are a lot of other databases. This NCBI is a complex database that have genome, that have nucleotide, that have protein. There are particularly protein database. In the next class, we will also do, we will try to understand microbial proteins. Why we need to understand them, we will study that also. There are polysaccharide database, there are lipid database, there is database of almost all kind of biological macromolecules. There are database of organisms also. There is database of E. coli, there is database of C. elegans, there is database of rice. So organism also have its own database. What is database? We deposit an information in a database, isn't it? So when you open that website, you get all the information related to that. So here you can see that our Bacillus subtilis is showing the similarity with this strain. So what we can do is, there are 100 sequence. We can basically select the first 10 sequences. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. And we go into distance tree of result. You can see their phylogeny also here. So you can see Bacillus subtilis, Bacillus licheniformis, our sequence is showing the similarity with Bacillus subtilis 10 JCM. You can, there are a lot of methods how you are making the phylogenetic tree. So in all the methods, they look a little similar. You can change the sequence difference also, and you can download it also. In the tools, you can download the phylogenetic tree. The same method will be applicable for almost all the bacteria. Just to try, let me change the bacteria to, okay. And if you want to see the sequence, when we are opening the bacteria, the 16S gene sequence, if we go to this option FASTA, there is the entire nucleotide sequences, you see. So this is the housekeeping gene that is common in all the Bacillus licheniformis that I was talking about. Normally 16S gene sequence have 1500 base. So almost it's a 1500 base long. Often it's a little less due to sequencing. You can lose a few bases due to errors, but it's nearly 1500 base pair. And you get all the information, when this was deposited, by whom this has been worked, all the details you will find in this part. We can search other bacteria, for example, Bacillus cereus. You can write 16S rRNA also. So there comes the 16S rRNA. There will be a lot. We can search and we can purify our search, removing the whole genome sequences, only the 16S gene. So if we click on this, we can see the information here. And if we click on FASTA, there comes the genome. So we can see it's a complete 16S, almost complete 16S gene sequence, rRNA. So if we go here in Run Blast, it will compare with the database of Bacillus. We have to first select rRNA database and let us go to Blast. So it will give you similarity with more than 100 bacterial species. How we can identify them? Look, the query cover is 100%. What is query cover? With the compared sequence, with the sequence that you are comparing, are 100% comparing. If the sequence is small, can be 99. You can see the e-value, and you can see 100% similarity with Bacillus cereus and Bacillus cereus. With Bacillus sanguinis, 99.93%, and it's a different species. This is an example how you can understand that when it's 100% similar, it's same bacteria. But when it's more than 97%, the genus is same, but the species become different. While one is sanguinis, one is cereus. So, for example, let us select maybe 10 and we go to distance tree of result. And there you can see that our Bacillus cereus strain is showing the similarity with other Bacillus cereus strain, Bacillus cereus CCM 2010. So we can again go into tools and download option. We can download it as a PDF file. So that's a simpler way how, I hope you can see here, how if we have a bacterial sequence through 16S genome sequencing, there are sequencing machines also. PacBio, Illumina, a lot of sequencers are now there in the medical laboratories, in the research laboratories, in the universities, in the institutes. And when we have the isolated DNA, we amplify them in a PCR, we sequence them, and then comes the role of bioinformatics to compare them and understand who they are. So when we understand what, how and which bacteria it is, we identify them, let us think in a different way. Today we will be ending up in this part because we will not make your brain like bursting with a lot of information, but just to understand what our next class will be, let us think about what are proteins. We all know proteins, but bioinformatics helps to understand protein, the structure. The entire protein structure you can identify through bioinformatics. So, uh, why we are giving example of microbial protein, but the same way that I will show you in the next class, you can identify a lot. You can identify the protein of plant, you can understand the structure of a protein of animal. The the techniques are same. You can apply it to different organism. We will give the example of microbial proteins, but you can think in a different systems also. And why it is important to understand protein? When we are nowadays, we are in a time where we are taking supplements. I was talking about nutraceuticals. We are in a time where protein is important, and microbial protein have a lot of industrial importance, biotechnological importance. For example, microbial protein, one enzyme called amylase, this enzyme is regularly used in clothing industry. When we get a sari, when we get a cloth, all the time it's starched, isn't it? If it's a cotton sari or a cotton cloth, it's starched. In the industry, for processing of starch of the cloth, we need an enzyme called amylase. And you know, the problem of amylase is very heat sensitive. So there comes the microbiology. Amylase is produced by microorganism, but if the microorganisms are growing in a hot spring, in a fumarole, in a very high temperature, they can produce enzymes that are thermostable. And why thermostable is important? Imagine our moms or our grandmoms, when they do the starching of the clothes in home, they the starch, rice starch, we can see also, starch only comes out in hot water. So the temperature stability is a very big factor for protein. The protein can be functional, highly functional, but if the protein don't have temperature stability, often these proteins are not suitable candidate for the industry. So this is the reason we need to understand, okay, our protein have this, we know the protein, we know their structure, but are they stable? If they are stable, let us know their function. So when they are structurally and functionally both are characterized, that time we can say that, okay, we know a lot about the proteins. In the last classes with the BiXs kids, we have published even a paper, a scientific paper with kids like you all, about understanding the structure and function of an enzyme called xylose isomerase produced by bacteria, that is used a lot in the food industry, in the syrup, in medicine industry, in sucrose syrup, in all these things. So imagine the work has been done by all of you like kids in your project, and the paper got published in a good international journal. We are also working in few other enzymes called keratinase. You know, the problem of keratin degradation is a big problem of hair, of horn. Keratin is a very strong protein, and in the industry it's very difficult to treat keratin. In the time to bioremediate the pollution, bacteria produce keratinase enzyme and they can degrade keratin. So, in order to understand which keratinase enzyme can be a good candidate, we need to compare the sequence, isn't it? Produced by a lot of bacteria. And how you can work in the laboratory with so many models. The easiest thing can be using the bioinformatic tools. You download all the sequence, you compare them in your computer, sitting in your home, and understanding their structure, their function, and who is showing the better structure function interrelationship. So how the protein actually looks like? They have four different structure: primary structure, secondary structure, tertiary structure, and quaternary structure. Okay. The primary structure, just let us think about a string, a wire. A wire can be a copper wire, a wire can be a steel wire. We have all seen about the wires, isn't it? So this is the primary structure. Insulin hormone in our body is an example of a primary structure of protein. So one after one amino acid, then another amino acid, then another amino acid, then another amino acid, one after another attached, bits on a string structure. So you have a wire. Think like you are playing with the wire, you was trying to make it as a string. In pen, we have seen the secondary structure, isn't it? String. We can make a string using a wire, isn't it? So this is a secondary structure. The string can be different. Next classes we will start try to study more in deep with them. They can be helix when it's more conjoined. It can be strand when loose, can be coils. And then when you have a string and you try to mold the string to give it another shape, this is tertiary structure. Tertiary structure example can be actin protein in our body, tubulin, albumin in the egg. They remain in tertiary structure. So when you have one tertiary structure protein, another tertiary structure protein, let us give an example of our own DNA polymerase enzyme that help our DNA to divide. This DNA polymerase is a quaternary structure. Hemoglobin is a quaternary structure where two tertiary structure protein are attached with one and another. With the help of bioinformatics tools, we can see all these structures and we can identify structurally all these structures: primary, secondary, tertiary, quaternary. And these are the things that we will be going to study in the next class. Okay, so there you have to think, you have to, this week you will study this part. You will let me know by mail or by the WhatsApp group if you are able to do the phylogenetic analysis of the bacteria or not, or the microorganism. It can be a simple thing. You can let me know what are the name of the good bacteria that you know. Let us make a homework this week. Name of 10 bacteria that you know. 10 bacteria each of you. In the next class, I want to see from you name of 10 bacteria that you know. Don't search anywhere. Just try to remember if you know the name of 10 bacteria or not. Maybe not genus and species, only genus, the way you know it. Let us make this homework. So, to end up today, it's difficult bioinformatics, but the greater the difficulty, the more is the glory when you surmount it, isn't it? So today probably it can be looking a little difficult, but the next week will be a little more easier. We will have a in-depth look into the application of bioinformatics in microbiology, and definitely this week you all are going to do the exercise trying to do. When I was I'm really not sure if when I was doing the hands-on, if you were trying to do in your own computer screen or not, try to do it. And the next week I want to see from all of you one phylogenetic tree of one bacteria. So we have two homework: name of 10 bacteria that you know, and one phylogenetic tree of one microorganism from NCBI. Hello?
Yeah, it was wonderful, ma'am. I hope they followed up. Leah, Aditya, everyone can speak up if you got today's point or not.
Please let me know if there is some problem. There can be questions also, silly questions that you are thinking or fearing if you will ask me or not, but please feel free. Because the questions that you are having today, probably you will have for 10 more years, believe me. If you don't ask today, probably you will have the doubts continuing. So feel free to ask me, because when I was in your stage, believe me, I used to have a lot of silly questions, and I used to ask it also to my teachers. Often they scold me, but I never stopped to ask. I can
Yeah,
see I can see someone. I can only see his face, but he's not asking anything. So I really don't know if he tried, if he understood or not, but I hope he had been. And I hope you all have tried at least the part of the hands-on when I was doing, because if you are not doing it by your hand, bioinformatics is such a thing that you have to do it in the computer. And the easiest thing is, we all are nowadays tech savvy. The kids are more tech savvy these days, and let us use this as our strong power. And this is the thing that you are using now. You can use it. We have a lot of kids. A good example can be Maurya. If they, I don't know, it can be good to even talk to them about the other groups also. And Maurya came from BIX's batch, and then she published a paper, and now she's in USA. I hope Rajul, you know, isn't it, about Maurya? And has been a very amazing BIX's kid we got. She always had questions. So I think in the next class I will get some questions from you all after you try in home. Naya was also there. I think I was talking about Naya because Maurya was in another group. Hmm. Yes. It's a WhatsApp group, yes. Hmm. And when you have done it, you can send me even in the mail kits. Feel free to send me the name of the 10 bacteria and the phylogenetic tree that you have done. If you are doing it, I will be able to understand. Like if you are able to understand the process, and if you cannot, please let me know that also, so that in the next class we can start from there. So you get one more day to understand the things. Thank you, Rajul, and thank you all. Thank you, Dr. Sandeep. Thank you all, kids. See you. See you.